Hey everyone, let's practice some MCQs on Hexose Monophosphate Pathway, also known as Pentose Phosphate Pathway. Question number 1 is the haptose ketose sugar formed by the chemical reaction in HMP Shunt. The haptose ketose sugar means the 7-carbon ketose sugar that is formed in the HMP Shunt. HMP also known as Hexose Monophosphate Pathway, and the other name is the Pentose Phosphate Pathway. Option A is cerohaptulose, B galactohaptose, C glucohaptose, D manohaptose. The right option is the 7-carbon keto sugar that is formed in the pentose phosphate pathway is cerohaptulose. As you can see here, shown is the pentose phosphate pathway. The cerohaptulose is formed here, which is a 7-carbon molecule. Cerohaptulose is formed by the trans reactions between cellulose 5-phosphate and ribose 5-phosphate. These both are apimers of each other. This is the structure of the cerohaptulose 7-phosphate. It consists of 1, 2, 3, 4, 5, 6 and 7 carbon. So that's why it is known as haptose, and it has ketose and it has the keto group attached in it. So that's why the haptose ketose sugar formed in this pathway, HMP, also known as Hexose Monophosphate Pathway or also known as Pentose Phosphate Pathway, this 7-carbon molecule is known as the cerohaptulose. Question number 2. Two important byproducts of HMP Shunt are A. NADH and pentose sugars, B. NADPH and pentose sugars, C. Pentose sugars and 4-membered sugars, D. Pentose sugars and cerohaptulose. The right option is NADPH and pentose sugar. As in the pentose phosphate pathway, all these options are correct, but except for the A option in which there is NADH. There is no NADH production in pentose phosphate pathway. But all these products are produced and formed in the pentose phosphate pathway. But the right option is B, NADPH and pentose sugars, because these are the important products that have a significance. As NADPH, it is produced in the first reaction and third reaction. In the first reaction and third reaction, there is the dehydrogenation reactions in which there is the formation of NADPH in first reaction and third reaction. NADPH, they are involved in the reduction of glutathione, and glutathione, then they are involved in the reduction of oxidative stress that is caused in the cells. So that's how NADPH are involved in the reduction of oxidative stress and prevent the cells from lysis. Similarly, the pentose sugars, they are also formed in the pentose phosphate pathway, and these pentose sugars are used for the synthesis of nucleotides. Question number 3. The 4-membered aldose sugar phosphate formed in the HMP Shunt pathway is, they are asking about the molecule that consists of 4 carbon atoms and has aldose group present in it in the pentose phosphate pathway. What is its name? A. Xylulose phosphate, B. Erythrulose phosphate, C. Erythrose phosphate, D. Ribulose phosphate. The right option is the molecule that consists of 4 carbon atoms and the aldose group is the erythrose phosphate. As shown here, this is the pentose phosphate pathway. Erythrose phosphate is formed here. This erythrose 4-phosphate that is formed by the trans reaction between glyceraldehyde 3-phosphate and cerohaptulose 7-phosphate. Between these, the trans reactions give rise to fructose 6-phosphate and erythrose 4-phosphate. If we see the structure of the erythrose 4-phosphate, this is the structure of the erythrose 4-phosphate. It consists of 1, 2, 3 and 4 carbon atoms. So that's why it is known as 4-membered and has the aldose group attached to it. So that's why this is aldose sugar. So the 4-member aldose sugar phosphate formed in this HMP pathway, or also known as pentose phosphate pathway, is known as the erythrose phosphate. Question number 4. Our body can get pentoses from A. HMP Shunt, B. Uramic acid pathway, C. EM pathway, D. TCA cycle. The right option is HMP Shunt. Our body can get pentoses from hexose monophosphate pathway, also known as pentose phosphate pathway. Here shown is the pentose phosphate pathway, in which the pentoses are formed is the ribulose 5-phosphate. In the form of ribulose 5-phosphate, the pentose phosphate pathway or HMP shunt is divided into two phases. This 1, 2, 3. These three reactions are oxidative phase, and then after ribulose 5-phosphate there is the non-oxidative phase of the pentose phosphate pathway. This ribulose 5-phosphate can be converted into ribose 5-phosphate, as ribulose and ribose, these are isomers of each other. Ribulose has a keto group while ribose has a aldo group. Then ribulose can be converted into xylulose, these are apomers of each other. So ribose can be converted into xylulose by the enzyme apomerase. So there are three types of pentoses that are present in the body that are produced through HMP shunt. This is the ribulose, ribose and xylulose. These three types of pentoses, and these pentoses can be used in the synthesis of nucleotides. As we know that ribose is the basic sugar unit of the nucleotide structure. Question number 5. The following enzyme is required for the hexose monophosphate shunt pathway. Which enzyme among the following? A. Glucose 6-phosphatase, B. Phosphorylase, C. Aldolase, D. Glucose 6-phosphate dehydrogenase. The right option is, as glucose 6-phosphatase, it does not belong to the HMP shunt. Only the glucose 6-phosphatase dehydrogenase. Only in these options this enzyme is correct. Glucose 6-phosphate dehydrogenase, also known as G6PD enzyme. This enzyme, and the very important enzyme, belongs to the HMP shunt and pentose phosphate pathway. In this pathway, the glucose 6-phosphate dehydrogenase is involved in the first reaction, in which as soon the glucose enters inside the cell, it gets trapped by the glucose 6-phosphate through hexokinase and glucokinase, and then glucose 6-phosphate as the first reaction. Glucose 6-phosphate, it is converted into 6-phosphogluconolectone, and there is the dehydrogenation reaction. That's why the enzyme is known as dehydrogenase. Glucose 6-phosphate, because it causes the dehydrogenation of the glucose 6-phosphate. That's why the enzyme name is G6PD, glucose 6-phosphate dehydrogenase enzyme. This is the reaction that it catalyzes. Glucose 6-phosphate is converted into 6-phosphogluconolectone with the help of the enzyme glucose 6-phosphate dehydrogenase, also known as G6PD, and there is a dehydrogenation reaction. Hydrogen is removed from glucose 6-phosphate and is taken up by NADP, and NADP is reduced into NADPH. Question number 6. Dehydrogenase enzymes of the hexose monophosphate shunt are A. NAD specific, B. NADP specific, C. FAD specific, D. FNM specific. There is only one carrier that is specific for the hexose monophosphate shunt, HMP shunt, also known as pentose phosphate pathway. This is the NADP specific. The enzyme dehydrogenases enzymes are specific for NADP. As just like other pathways where sometimes NAD is the carrier and FAD is the carrier and sometimes FMN is the carrier, but in this HMP pathway and pentose phosphate pathway, the only carrier is the NADP. NADP get reduced into NADPH. As shown here, there are two types of enzymes in the oxidative phase of the pentose phosphate pathway, in the first reaction and the third reaction. The two types of dehydrogenases enzymes are used. One is the glucose 6-phosphate dehydrogenase, also known as G6PD, which causes the dehydrogenation reaction and converts the NADP into NADPH. The other dehydrogenases enzyme is the 6-phosphogluconolectone dehydrogenase. It also causes the dehydrogenation reaction. It converts the NADP into NADPH. And the other specific is that it also removes one carbon in the form of carbon dioxide. So two types of dehydrogenation enzymes are involved in this pentose phosphate pathway. One is glucose 6-phosphate dehydrogenase and another is 6-phosphogluconolectone dehydrogenase. And these both are involved in the dehydrogenation reaction, and it reduces the NADP into NADPH. So there is no NAD, no FAD, only the NADP is involved. NADPH, NADP will be reduced into NADPH, and NADPH they then reduce the glutathione, and glutathione then reduces the oxidative stress of the cell and prevent the cell from lysis. Question number 7. One molecule of glucose gives how many molecules of carbon dioxide in one round of HMP shunt? A. 6, B. 1, C. 2, D. 3. So the right option is 1. There is only one carbon dioxide projection in whole HMP and pentose phosphate pathway, and this one carbon dioxide is produced or formed in the oxidative phase third reaction. In this third reaction, as the glucose is the 6th carbon molecule, when it goes through reactions, then this is the reaction in which 6-phosphogluconeolactone is converted into ribulose 5-phosphate. So this 6-carbon sugar is converted into 5-carbon sugar to give rise to the production of pentoses, which are 5-carbon molecule. So this reaction is catalyzed by the enzyme 6-phosphogluconeolactone dehydrogenase, and there is the dehydrogenation reaction in which NADP is reduced into NADPH and one carbon is removed in the form of carbon dioxide. So 6-carbon molecule is converted into 5-carbon molecule, which are known as pentoses in the form of ribulose and ribose, and which are then used for the synthesis of nucleotides. Question number 8. Reduced glutathione functions in RBCs to A. Produce NADPH, B. Reduce methemoglobin to hemoglobin, C. Produce NADH, D. Reduce oxidizing agents such as hydrogen peroxide. The right option is D. The reduced glutathione in RBCs is used to reduce oxidizing agents such as hydrogen peroxide. As the reduced glutathione is reduced by NADPH. This NADPH, as it contains the hydrogen, so this is the glutathione in oxidized state. This oxidized glutathione gets reduced by NADPH, and we know that NADPH, it is reduced by the enzyme glucose 6-phosphate dehydrogenase, also known as G6PD. G6PD enzyme, glucose 6-phosphate dehydrogenase enzyme, it reduces the NADP into NADPH, and then NADPH, it reduces the glutathione into reduced state. And this reduced glutathione, then it breaks the harmful free radicals in the form of HgO2 into water and oxygen. So reduced glutathione, it reduces the oxidation as superoxide radicals, supermutase and hydrogen peroxide, and converts them into less toxic water, as water is non-toxic. So that's how it prevents the cells from toxic free radicals and it prevents the cells from lysis. Which of the following enzyme deficiency makes individual resistant to malaria disease? A. Transaldolase, B. Transketolase, C. Glucose 6-phosphate dehydrogenase, G6PD, D. All of the above. The right option is C. Glucose 6-phosphate dehydrogenase. This is the enzyme which deficiency makes the individual resistance to malaria disease. How the individual gets resistance to malaria disease in which this G6PD enzyme is deficient? What is the reason behind it? As G6PD enzyme, which is formed in the first reaction of the pentose phosphate pathway, glucose 6-phosphate is converted into 6-phosphogluconeolactone. This G6PD enzyme generate NADP into NADPH, and this NADPH, as previously I explained how NADPH reduces the oxidative stress, that it reduces the glutathione, needed for reduces glutathione, it reduces the glutathione, and glutathione then maintains the RBC's membrane. How? By oxidizing, by removing the free radicals and reducing the hydrogen peroxide into non-toxic water molecules. So that's how it maintains the RBCs and prevent them from lysis. So if there is malarial parasite, as this G6PD maintains the RBCs from oxidative stress, whenever there is malarial parasite, it gets into the RBCs, then it makes the malarial parasite to survive difficultly. Then due to difficult survival, because of the increased oxidative stress, So that's how the parasite may not be able to reproduce and replicate. So that's why it protects from malaria diseases. The malaria parasite Plasmodium vivax and falciparum invades and replicates within the red blood cells. As malaria parasite, they inhabit and enter into the red blood cells and they feed on the red blood cells. Red blood cells from individuals with G6PD deficiency are more susceptible to oxidative stress. So the individuals having deficient in this G6PD enzyme, then there will be no reduced glutathione and there will be increased oxidative stress. The increased oxidative stress in G6PD deficient red blood cells can disrupt the parasite's ability to invade, grow and replicate. So that's how when the oxidative stress in the RBCs are increased, it will disrupt the parasitic growth and replication. Enzyme deficiency that leads to the development of hemolytic anemia in patients treated with antimalarial drugs. A. Transaldoles. B. Transketolase. C. Glucose 6-phosphate dehydrogenase. D. All of the above. So they have asked about when the patient is given antimalarial drugs and then it causes hemolysis, which may be likely due to deficiency of the enzyme glucose 6-phosphate dehydrogenase G6PD enzyme deficiency. If these patients and peoples are given antimalarial drugs, their cells may lead to the hemolysis. This is because, as I previously explained, G6PD enzyme, it is used for the dehydrogenation, for the reduction of NADP into NADPH, and NADPH reduces the glutathione. And then reduced glutathione, it reduces the oxidative stress of the cells. So when G6PD enzyme is essential for red blood cells to function properly, as I told, that it maintains the RBC's membrane by reducing the oxidative stress. Individuals with G6PD deficiency have red blood cells that are more susceptible to oxidative stress. So G6PD deficiency patients will be more susceptible to oxidative because they have deficient in G6PD enzyme, so they will be having increased oxidative stress. When these individuals are exposed to certain medications, including some antimalarial drugs, their red blood cells can be damaged and destroyed prematurely, leading to hemolytic anemia. So these patients which are already due to G6PD deficiency, their cells are high in oxidative stress, and when they are given antimalarial drugs, then they will cause the lysis of the red blood cells. It will again enhance and destroy and damage the RBCs because of the increased oxidative stress. This was one of the examples of the antimalarial drugs. There are some other options that may be asked in the MCQs. You must know about other hemolytic factors. What are other? Because this is the only one drug I have mentioned here. There are also some other medications which may lead to the hemolysis of the red blood cells. These hemolytic factors are fava beans. There are some people that are allergic to fava beans. When they ingest these fava beans, because they are deficient in G6PD enzyme, and when they ingest these fava beans, then they increases the oxidative stress of RBCs and they lead to the hemolysis. Similarly, there are some other medications like primaquine, chloroquine, aspirin, even aspirin, sulfonamides, nitrofurantoin, and similarly there are certain infections. There are some certain chronic diseases and infections, inflammations, in which also causes the increased oxidative stress, and if there is G6PD deficient, then there will be no compensatory mechanism to reduce the oxidative stress, and that will lead to the hemolysis and disruption of RBCs. Question number 11. Rearrangement reactions unique to the HMP pathway are catalyzed by the enzymes A. Transaldoles, B. Transketolase, C. Both A and B, D. Pentose phosphate epimerase. As shown here is the pentose phosphate pathway. As pentose phosphate pathway, HMP pathway has two phases, oxidative phase and non-oxidative phase. They are asking about non-oxidative phase. In non-oxidative phase, the rearrangement reactions are here. In fourth reaction, fifth reaction, and then sixth reaction. There are the rearrangement reaction, or we say that trans reactions occur here. These reactions takes place with the help of the enzyme as shown here, transketolase and trans-aldolase. These are the trans-ketolase and trans-aldolase. So the right option is C, both A and B, trans-aldolase and trans-ketolase. Trans-aldolase that transfers the aldo group, trans-ketolase that transfers the keto group. So trans-ketolase and trans-aldolase that are involved in the transfer reaction rearrangement, in which there is the 5-carbon sugar and 5-carbon sugar, and then they transfers the groups and carbons, then it forms the glyceraldehyde 3-phosphate and 7-carbon molecule 0-heptulose, and then again there is the transfer of aldehyde group, then it forms the fructose 6-phosphate, which can go to the glycolysis, and erythrose 4-phosphate, which further involved in the reaction with xylulose 5-phosphate and gives rise to again fructose 6-phosphate and glyceraldehyde 3-phosphate. Both are the intermediate product of the glycolysis. So in simple words, rearrangement reactions takes place by trans-ketolase, trans-aldolase, trans-ketolase. So these two types of enzymes are involved in the rearrangement reactions in the non-oxidative phase of the pentose phosphate pathway. Glucose 6-phosphate dehydrogenase enzyme is negatively regulated by A. Increase ATP, B. Increase NADP, C. Increase NADPH, D. Increase AMP. The right option is glucose 6-phosphate dehydrogenase is negatively regulated by increased NADPH levels. When there is enough production of NADPH, it will negatively, by feedback inhibition, it will inhibit the activity of the glucose 6-phosphate dehydrogenase enzyme. As this is the first reaction of the pentose phosphate pathway, in which glucose 6-phosphate converts into 6-phosphoglucose electron with the help of the enzyme glucose 6-phosphate dehydrogenase. There is the dehydrogenation reaction in which NADP is reduced into NADPH. So when there is increased and enough production of NADPH, it inhibits the glucose 6-phosphate dehydrogenase enzyme activity. So this is the negative feedback inhibition. G6PD is negatively regulated by NADPH through a feedback inhibition mechanism. So this is known as the feedback inhibition mechanism. NADPH levels increase, it binds to the enzyme and inhibits its activity, slowing down the production of more NADPH. So when there is increased production of NADPH, it will bind to the enzyme and slow down the more production of NADPH. This helps maintain a balance of NADPH levels within the cell. So that's how the level of NADPH level are balanced inside the cell. Question number 13. Glucose 6-phosphate dehydrogenase enzyme is positively regulated by A. Increase ATP, B. Decrease NADP, C. Increase NADPH, D. Increase AMP. The right option is increase ATP. As increased ATP synthesis, there is increased demand of cellular energy levels, so due to which it will require molecules for their activity. This is the first reaction in which glucose 6-phosphate is converted into 6-phosphoglucose electron with the help of the enzyme G6PD, in which there is the synthesis of NADP into NADPH. So why the G6PD is positively regulated by increased level of ATP? When cellular energy levels are high, as indicated by increased ATP levels, so it will activate more pathways that require NADPH, such as fatty acid synthesis and nucleotide biosynthesis. So increased ATP levels and high energy levels, it will demand more synthesis of fatty acids and nucleotide biosynthesis, so in which the NADPH is required for the synthesis of fatty acids and nucleotide biosynthesis. This increased demand for NADPH can indirectly stimulate G6PD activity. So for the increased amount of NADPH, it will activate the G6PD to synthesize NADPH, and which will be utilized for the synthesis of fatty acids and nucleotide biosynthesis. As previously we discussed about that NADPH is involved in the reduction of glutathione, but the other functions of NADPH are fatty acid synthesis, and they are also involved in the nucleotide synthesis. Question number 14. Pentose phosphate pathway occurs in A. Mitochondria, B. Nucleus, C. Cytoplasm, D. Golgi apparatus. The right option is pentose phosphate pathway occurs in cytoplasm of the cell. As this is the pentose phosphate pathway, and this occurs in the cytoplasm of the cell, as like glycolysis also takes place in the cytoplasm, while the Krebs cycle and electron transport chain, it takes place inside the mitochondria. To evaluate the vitamin B1 nutrition deficiency status in a patient, measurement of which of the following can be used: A. Erythrocyte transcellulase, B. Erythrocyte transketolase, C. Glucose 6-phosphate dehydrogenase, D. Glucose 6-phosphogluconate. The right option is erythrocyte transketolase, because the transketolase enzyme that is used in the Pentose Phosphate Pathway HMP Shunt, this enzyme transketolase, it has the prosthetic group thymine. So that's why erythrocyte transketolase is used to determine the deficiency of vitamin B1. The test is known as erythrocyte transketolase activity assay, in which thymine level is determined, and then if the thymine is deficient, then the supplementation or nutritions are recommended. When thymine levels are low, the activity of transketolase, particularly in red blood cells, decreases. So the low thymine level indicates the low level of transketolase. Then the supplementations and the nutritions are suggested to overcome the deficiency. Thank you for watching, and keep watching to learn more.